Welcome to Cancer Lesson 2.2, How is Replication Controlled in a Normal Cell and Disrupted in Cancer? The goals of this lesson are to introduce how cell division is controlled through the cell cycle, and explore how each step of the cell cycle works and how cell division can become uncontrolled. At the end of the lesson, the students should be able to describe the significance of interphase during the cell cycle, explain how driver proteins move the cell cycle forward, describe how checkpoint proteins inhibit cell cycle progression, and explain how drivers and checkpoints can lose control of the cell cycle, leading to unregulated cell growth. We will achieve these goals through a Socratic discussion and by engaging in a board game simulation of cell cycle progression. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. The process of cell division is called the cell cycle and is controlled by signals that either push the cell from one stage to the next or stop the cell dividing. If these signals are abnormal or not recognized, cells either divide uncontrollably or die. In cancer, these signals have lost the ability to control the cell cycle. As a result, cancer cells divide uncontrollably. You can review this scientific information in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual, or lesson plan, provides a minute-by-minute explanation of lesson structure. The student workbook provides additional explanation for students. Be sure to print the lesson worksheet and the homework worksheet. You will also need the cell cycle game boards and the cell cycle game dice. The key points of the do now are that mitosis is only a small part of the cell cycle. Most of the time is spent in interphase, preparing the cell to divide. Start and stop signals control how cells pass through different parts of interphase. The students should understand that mitosis is only a fraction of what cells are doing during their life cycle. In fact, during most of the cell cycle, the cell is either preparing to divide or pausing while it waits for a signal telling it to proceed. This part of the cell cycle is called interphase. Interphase is necessary because regulating when a cell will divide is critically important either to keep the tissue in homeostasis or to repair it after injury. Normal cells know whether to start or stop mitosis because of signals they receive during interphase, which act like traffic lights. Start signals tell the cell to continue through the cell cycle to mitosis, while stop signals halt it at certain points in the cell cycle. These start and stop signals are provided by the environment of the cell. The key points of the discussion are that start signals tell driver proteins to push the cell into the next phase of the cell cycle, and stop signals tell checkpoint proteins to inhibit the driver proteins. We can think of the cell cycle like a clock, with gears that move the cell towards mitosis. The first important concept is that the signals controlling the cell cycle are found in the cell's nucleus. The cell cycle clock is divided into different phases. Use the animations on this slide to demonstrate how driver signals move the cell from one phase to the next, while the checkpoint signals pause the cell in a specific phase. The second important point is that driver and checkpoint signals are responsive to the environment. Next, we zoom into each of these phases, starting with the G1 or gap 1 phase. Here the cell is looking for signals from the environment to determine what it should do. If it receives start signals, it will move on to the S phase or synthesis phase. Here the cell duplicates its chromosomes. Next, cells go into the G2 or gap 2 phase. Not very much is known about the details of the G2 phase, but we think that during the G2, the cell is preparing for mitosis, which is when the cell divides into two daughter cells. Next we ask, how does the cell know when to go from one phase to the next? A series of proteins act as traffic lights. Driver proteins are the green lights that tell the cell to move from G1 to S to G2 and then on to mitosis. Checkpoint proteins are the red lights that prevent the cell from moving on to the next phase. Checkpoints act by interfering with the drivers. Both drivers and checkpoint proteins are made in response to cues from the environment. Use the animation to illustrate that each phase of the cell cycle has its own driver that needs to become active in a specific order for the cell to progress to mitosis. We can draw a graph that illustrates how the actual amount of each driver protein fluctuates over the course of the cell cycle. Notice how the amount of each driver on the vertical axis increases to a maximum during the phase it controls and then decreases. But what exactly are these drivers? Each driver protein is in two parts. The first part is a switch, the second is called a cyclin. The cyclins need to be switched on in order to move the cell to the next phase of the cycle. Checkpoints are the red traffic lights of the cell cycle that tell the cell not to progress to divide at this moment in time. There are many reasons why a cell should not divide. For example, there may not be enough nutrients available, or the parent cell may not have finished duplicating its chromosomes. There are 3 cell cycle checkpoints that are important in cancer, so we will spend some time on them. At each of these 3 checkpoints, the cell either samples the environment or checks on its own status and then decides whether to continue towards mitosis. Animate the slides to step through them. The checkpoint at the end of G1 asks whether the environment will support cell division. Are there enough nutrients? Is there room for more cells? It also asks whether the cell's DNA is intact and can be replicated properly. The checkpoint at the end of G2 asks whether DNA replication has been successful. The checkpoint in the middle of mitosis asks whether the chromosomes are ready to be separated. The checkpoint at the end of G1 is particularly important because after this point, the cell will progress through the cell cycle even if external signals to tell it to stop appear later. It is called the restriction point. Checkpoints stop cells passing through the cell cycle by interfering with the drivers. Each phase of the cell cycle has its own checkpoint protein. This checkpoint can attach to the driver that is active during that phase and stop it from working. The full set of slides is animated to illustrate how each checkpoint protein inhibits its driver. Checkpoint proteins are very important because they stop cells dividing when external conditions are unfavorable or when the cell is damaged. Next, we take a closer look at 2 important checkpoint proteins that work in different ways. The retinoblastoma checkpoint is often known as RB. It is particularly important because it works on the G1 driver at the point of no return, the restriction point. Think of RB like a barrier at a parking lot. It prevents the cell passing the point of no return until it has fed in the ticket saying it's okay to proceed. The INK, or inhibitor kinase proteins, make sure that a cell doesn't actually divide until everything is in order to make sure the daughter cell will be normal. INK proteins work on the G2 and mitosis drivers. The activity for this lesson is a game that simulates how cells progress through the cell cycle. The key points of the activity are that it is hard for cells to divide because they won't progress through the cell cycle unless each driver is activated in a specific sequence and each checkpoint has been passed successfully. Cell division is under such strict control so cells with DNA damage aren't replicated. In the cell cycle game, students work in groups of 3. 2 students play epithelial cells that must activate all the drivers in the appropriate order to progress through the cell cycle and divide, while the 3rd student provides signals to tell the cells to divide or not to divide. Further instructions on how to play the cell cycle game may be found in the worksheet. The key points of the wrap-up are that cancer starts off as hyperproliferating cells. Hyperproliferation can occur if cell cycle control is lost. This can happen if cells ignore surrounding signals, have drivers always active, or get rid of INK or RB proteins. DNA damage leading to mutations can also lead to loss of cell cycle control. For example, if cell cycle drivers are mutated to be always active and cell cycle checkpoints are mutated to be always non-functional, cells will hyperproliferate. The first point of the wrap-up asks how one can cheat in the cell cycle game to increase the ability of the cells to divide. Uncontrolled mitosis is known as hyperproliferation. Next, we think about the causes of hyperproliferation. We can think of the cell like a car. Why would a car suddenly start to go too fast? One way may be by pressing too hard on the accelerator, or if the accelerator was stuck on. Another way may be by not pressing the brakes hard enough, or if it was broken. Too many divide signals is analogous to pressing too hard on the accelerator. Not enough don't divide signals is analogous to not pressing on the brakes hard enough. A stuck accelerator or broken brake is analogous to a defective driver or checkpoint protein. Next, we think about what happens if the car loses control. Drivers and checkpoint proteins can become defective if they are mutated. The car going too fast is analogous to hyperproliferation. We will use this car analogy a lot going forward. Make sure students are comfortable with it. The last slide of the wrap-up reminds students to think about the vocabulary they learned earlier. They should realize that drivers are proto-oncogenes and control the cell moving forward in the cell cycle, and that checkpoints are tumor suppressors and control the cell pausing in the cell cycle. For homework, have students read an informational sheet from the Cancer Research UK on how chemotherapy kills cancer cells based upon the fact that cancer cells are hyperproliferating. One of the most common challenges students have is, do cancer cells progress through the cell cycle faster than normal cells? Cancer cells progress through the cell cycle at the same rate as their parent cell. However, what is abnormal is that their division is uncontrolled. At the end of the lesson, collect the lesson worksheets to assess students' explanations of the functions of drivers and checkpoints in the cell cycle. This lesson introduced the cell cycle and discussed some of the signals that control cell division. Next, we will look more closely at the environmental signals that affect the cell cycle. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.